What if the common understanding of DNA and evolution was challenged? What if there was more than one code for DNA? For years, it has been taught that there is a universal DNA code, a common language that all life shares. This universality was often cited as evidence for evolution. However, the National Center for Biotechnology Information, or NCBI, currently acknowledges not just one, but 26 different coding languages for DNA. And if that wasn't enough, there are also known to be a plethora of codes which exist outside of DNA. Codebiology.org currently lists 24 additional codes which exist outside of DNA. The living world teems with organic codes, and among them, species-specific splicing codes are of particular interest. In a 2012 article titled "Evolution by Splicing: Comparing Gene Transcripts from Different Species Reveals Surprising Splicing Diversity," it is revealed that there is a large amount of species-specific alternative splicing. Most alternative splicing events differ widely between even closely related species. The alternative splicing patterns are very different even between humans and chimpanzees. Through alternative splicing, one gene can encode more than one protein, with numbers reaching up to 1,000. The human genome contains about 21,000 protein-encoding genes, but the total number of proteins in human cells is estimated to be between 250,000 to 1 million thanks to alternative splicing. Moreover, alternative splicing can produce variant proteins and expression patterns as different as the products of different genes. In an article titled "Widespread Expansion of Protein Interaction Capabilities by Alternative Splicing," it is stated that alternatively spliced isoforms of proteins exhibit strikingly different interaction profiles and thus, in the context of global interactome networks, appear to behave as if encoded by distinct genes rather than as minor variants of each other. As many as 100,000 distinct isoform transcripts could be produced from the 20,000 human protein-coding genes, collectively leading to perhaps over 1 million distinct polypeptides obtained by post-translational modification of products of all possible transcript isoforms. The implications of species-specific splicing codes on our understanding of evolution are profound. As Raymond Bolin stated, where chimps and humans really differ, and they differ by orders of magnitude, is in the genomic architecture outside the protein coding regions. They are vastly different. The structural, the organization, the regulatory sequences, the hierarchy for how things are organized and used are vastly different between a chimpanzee and a human being in their genomes. In short, species-specific alternative splicing codes and gene regulatory networks in general challenge the very basis of the theory of evolution. As the 2018 article "Developmental Gene Regulatory Networks: An Insurmountable Impediment to Evolution" stated, a gene regulatory network, DGRN, that regulates body plan development is very impervious to change and usually leads to catastrophic loss of the body part or loss of viability altogether. This observable consequence virtually always occurs if even one DGRN subcircuit is interrupted. Because most of these changes are always catastrophically bad, flexibility is minimal, and since the subcircuits are all interconnected, there is only one way for things to work. And indeed, the embryos of each species can develop in only one way. To sum up, the discovery and understanding of species-specific splicing codes and/or gene regulatory networks show us that there is a vast diversity and complexity to life's codes that are species-specific. Splicing patterns and regulatory networks reveal vast differences in protein expression, even from a single gene. In conclusion, these species-specific codes are overturning universal common descent, which is arguably the very basis of the theory of evolution.